All right. Anyway, all right. Let's move on. Freeberg, you have a science corner.
I was going to share this was last week. I think we talking about it this week. So there was a paper that is a pretty compelling paper published by a team led out of Harvard on identifying what may be the core driver of aging and demonstration on an ability to kind of reverse aging. So I'll just start really quickly that, in the human body, we have many different types of cells, right? We have 200 roughly different kinds of cells: an eye cell, a skin cell, a brain cell, a heart cell. They all have the same DNA, the same genetic code, the same genome at the nucleus of that cell. What makes those cells different, and the reason they act and behave differently, is they have different gene expression, meaning different genes in that cell are turned on and off. And when a gene is turned on, the protein that gene codes for is expressed and made in the cell. And the genes that are off, those proteins are not made. And remember, proteins are the biochemical machines in biology. So when certain proteins are produced, they do stuff and other proteins don't do stuff, and the cell acts and behaves very differently. So some cells, when you turn genes on and off, you get a neuron. Some cells, you turn them on and off, you get a muscle cell in your bicep. Some of them, you get a heart cell. And so all of these cells are differentiated by the genes that are expressed. The general term for the expression of genes is the epigenome. And an epigenome basically refers to these systems whereby there's certain parts of the DNA, certain segments of genes are uncoiled a little bit. So if you zoom in on DNA, there's 23 chromosomes. They're tightly wrapped in these coils. And when you go even closer, you see that there's these segments called nucleosomes. And a nucleosome means it's like a bead, and a bunch of DNA is wrapped around the bead. And how closely those beads are together, how much the DNA is wrapped, allows a segment of the DNA to be opened up and then expressed, meaning copies of the DNA are turned into RNA, which floats into this thing called the ribosome, and the ribosome is the protein printer. So the more these little segments of genome are exposed, the more they're expressed. And there's certain chemicals, these methyls and acetyls, that kind of attach to the genome, and certain elements that allow parts of the chromosome to wrap up and get really tightly bound, or to unwrap and to express the gene. So the epigenome is almost, you can think about it like the software, and the genome, or the DNA, is the hardware. And so the hardware basically defines what you can make. The epigenome defines what is being made, what stuff is turned on and what stuff is turned off. So this paper and this work that was done, historically, we've always thought that aging meant that over time, the DNA in our cells was mutating and errors were accumulating in the DNA. And as a result of those errors, the cells start to dysfunction. And what these guys really did a good job of proving with this paper is that it may not be mutations in the DNA that's causing aging, but actually changes in the epigenome, and that the DNA remains pretty stable and pretty consistent over time. And the way they did this is they broke the DNA. And just so you guys know, every second of your life, about 1 million breaks in DNA in cells throughout your body are happening. Your DNA is being broken up, and then there's all this machinery in your cell that fixes the DNA when it breaks. Now, what happens when it fixes it, it turns out it's actually really good at fixing it, and the DNA doesn't change. And we historically thought that the DNA changed a lot, and mutations accumulate over time. But in reality, what may be happening is as your DNA gets fixed, the epigenome, the acetyl and methyl groups on the gene, on the chromosome, don't get put in the right place. And over time, what happens is the epigenome degrades. And this is considered, and a lot of people refer to this now as the information theory of aging. You can kind of think about making a lot of copies of software, a lot of copies of a photo in a photo printer over time. And every time you make a copy, there's a little error, and those errors accumulate. And the errors that accumulate cause the epigenome to change. And as a result, certain genes are turned on that are supposed to be off, and certain genes are turned off that are supposed to be turned on. And then those cells start to get dysfunctional because the wrong proteins are being made and the cell can no longer do what it's supposed to do. So what these guys...
Could it be the ribosome as well that gets screwed up over time, the printer?
The ribosome is a pretty, static protein. It just does its thing. And there's hundreds of ribosomes in a cell. So, if one of them's dysfunctional, it just doesn't do anything. And then the other ones kind of step in and do it. So the ribosomes are constantly running. What these guys did is they basically took two populations of mice, and they gave the one population of mice a certain thing that caused its DNA to break at 3 times the rate of the other population. And then as the DNA broke, they could they could see that this mouse population got older and older faster, and by a bunch of measures on how do you measure age. But what they did is they then sequenced the DNA of the two populations of mice. And what they showed is that the older mice, the ones that had their DNA changing a lot, by the way, these were genetically identical mice, the ones that had their DNA broken a lot more, they had the correct genome. Their genome was the exact same as the other mice that stayed young. And so what that tells us is that it's the epigenome and not the DNA itself that's changing. So then here's what they did. Remember last year we talked about Yamanaka factors, which are these 4 proteins, these 4 molecules that can be applied to DNA, to a cell, and they cause all of the gene expression to reset back to looking like a stem cell. And remember, all of those differentiated cells come from a stem cell. And when they did that, the older population of mice suddenly started to act younger, and all of the measures of age reversed. And they did this across different tissue types. They measured this in a lot of different ways: cognitive function, health, cellular health, etc. And so it is not just a fantastic new proof point of how Yamanaka factors can actually reverse age, but it demonstrates that the epigenome itself is what is the core driver of aging. And you guys remember Altos Labs raised $3 billion in a seed round last year. And you remember at the end of 21, I said like Yamanaka factors and aging research is going to be kind of the next hot thing. I think this paper is going to be one of the seminal papers that really kind of illustrates and proves the point that this epigenome is the driver of aging. And as we now are investing a lot of money in figuring out how Yamanaka factors and other transcription factors like the Yamanaka factors can be applied in specific ways to actually reverse aging and cause the cells to start functioning correctly again. And then people will start to act and resolve in a healthy way once again. There's a lot of work to go between here and there, but now we have a much more kind of definitive proof point that this information theory of aging may be real, that it's tied to the epigenome, and that there are solutions that can work. And we have to figure out how to put them together and how to engineer a fantastic outcome. So really great paper by a team led out of Harvard. I think really validates a lot of the work and the money that's going into the space, both in the public and the private sector. And obviously a lot of new startups kind of chasing this opportunity to figure out how we can use these transcription factors to reverse aging, and that this may end up leading us to a much kind of healthier life. And by the way, when they applied those Yamanaka factors to the mice lived 107% longer than they were supposed to. But more importantly, the health span, as it's defined, improved. So the mice not only lived longer, but they actually lived healthier. All the measures of healthiness in the body improved. So it's a really kind of powerful indicator.
Is this going to help us in the next 10 years?
It may. Yeah, it very well may. There are now some therapeutic efforts.
Is that how long they gave you Sachs? They gave you
Science Corner. I feel like Science Corner should only discuss things that can happen in the next few years. Let's put it that way. Or is that the wrong way to look at it?
I'll tell you one thing for sure. You can make money as an investor over the next 10 years in finding the right teams that are going to have the highest likelihood of progressing clinical trials in this space. I will say that there may be clinical trials that can come to market really fast, particularly with kind of these ex vivo therapeutics where you take cells out of your body, apply the Yamanaka factors, and then put them back in your body for certain tissue types, like eye cells, for example, or T cells in your blood. There's a lot of ways that this may come to market faster. And it's not just about reversing your age overall, but reversing the age of certain cell types in your body that can then have profound health impacts in the near term. So that's the kind of stuff that's going to start to come through clinical stage sooner than later. And then maybe, some number of years down the road, we figure out a way to reverse the age in all the cells in our body, and the whole body becomes more youthful. But for now, it's going to be targeted cells in a very specific way to reverse aging and improve health. Very powerful, very interesting, lots of investment opportunity, and certainly some very smart folks.
So what do you think the realistic time frame is for reversing aging? Because we need that.
30 years.
30 years? Shit.
Yeah. But I think
I'll be 80.
That's when you're going to need it the most. No, but that's when you'll need it the most.
But so Freeberg, seriously, if we live to 80, does that mean we're going to be able to live to 100 because we'll be able to reverse age?
Well, it depends. I think the question is, can you live well to 100? I think that's the question. We don't know.
But if you could reverse aging.
Yeah, but we don't know what that means, because there's all kinds of things that you inherit over time that this may not. For example, like if you have long-term heart disease, I could see how the cells could get healthier, but it can't eliminate the plaque in your arteries.
Right.
That's totally hardened calcium.
That's
so
right.
that shit's there. So you have to live well.
Same with Alzheimer's. Like Alzheimer's has there's a plaque element, but the cause of that and the cellular dysfunction may be reversible.
It could definitely be that injury rates of older people, hips, knees, shoulders, arms, all the sort of like soft musculoskeletal stuff, you can really do a good job of, because at the same time as you get older, like people intake less protein, they process it less well, you lose a lot of muscle mass as you get older. Those are things that I think are like short-term solutions. But no, to be honest with you, Sax, the stuff that really can screw you, which is heart disease and brain function, this probably won't do much for a long time.
So JCal's screwed.
Yeah. I'm in the best shape I've been in 20 years. I feel great. Can
you reverse whatever is wrong with JCal, or is that in the plaque category?
Can I do a quality of life shout out? Can I do a quality of life shout out?
Please. Yeah.
I got an email. We all got it from a guy who I won't say just not to violate his privacy. But he's in Saskatchewan. He listens to the pod, or his father is, and made him get a pre-nuvo scan.
Oh,
flew the father to Vancouver. They found a 5-centimeter cancerous tumor on his kidney, and 3 days ago had it removed, and looks like guy's totally healthy and eliminated. He was saying so. Another life saved.
On the pod.
But I wanted to show you guys a picture. So yesterday I went to Los Angeles to see my interventional cardiologist. And what they do is they do what's called a contrast CT scan. So they put you into an IV and they put this contrast inside of you. Nick, do you want to throw the picture up, please? And then they use all the software to actually create an extremely accurate 3D model of your heart. And what they can do is go inside of your arteries and actually measure the calcium buildup. I've mentioned this before. This is a service called HeartFlow, HeartFlow. In any event, my calcium score is still 0. Thank God, touch wood, keep grinding. But I just wanted to put this out there for anybody who has a history of heart disease in their family, for them or for their parents or what have you. If you go and ask your doctor, this is a third-party service that they can do it. You go get a contrast CT, and you can get a very accurate sense of your heart health.
This is amazing. They found
it's incredible.
They found that you have a heart.
They did find that I have a heart.
This is incredible technology, because all of us, we've been trying to find if you had a heart for all this 112 episodes.
Dr. Carlsberg was shocked.
Breaking news. They found a heart.
It looked huge. It looked like Secretariat's
Looks
heart.
like he's got a big heart.
Yeah.
I do have a big heart, boys, as you guys know.
No, I mean, this is shocking for the audience.
Is that actual size? That can't be actual size.
His heart's bigger than his brain. He's got a big heart. Well, I'm glad you're healthy, bestie. That's fantastic.
So go get a HeartFlow if anybody has heart disease. Go talk to your doctor.